The broadcast is now starting. All attendees are in listen-only mode.
Hello everybody, and thank you for joining us today for the Nelson Labs webinar, History and Modern Practices for ASTM E1174, Evaluating the Effectiveness of Healthcare Personnel Handwash Formulations. This webinar will be presented by Christopher Beausoleil. More on him in a moment after a brief overview of our company. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 900 microbiological and analytical laboratory tests. At Nelson Labs, we know that every test matters. If you would like to watch any of our recorded webinars, including today's, you can find them on the Nelson Labs website listed under Education in the main menu. You can also learn about upcoming live and on-demand webinars by following us on LinkedIn or checking on the Education and News and Events sections of our website. We also invite you to register for the Sotera Health Academy at academy.soterahealth.com. This free educational resource offers curated content from industry thought leaders within the Sotera Health family. From introductory sterilization and lab testing topics to advanced and in-depth material, our expert advisors have filled this academy with cutting-edge industry content and resources to help you mitigate risk, get to market faster, and achieve excellence in your field. Now I'll introduce the presenter of today's webinar. Chris Beausoleil is an accomplished department scientist at Nelson Labs supporting our Bozeman, Montana facility. Since joining Nelson Labs Bozeman in 1994, he has a career spanning over 30 years in all phases of clinical research and antimicrobial efficacy evaluation. As a key resource for sponsors, he specializes in conducting complex clinical studies, focusing on evaluating the efficacy and safety of products using standard FDA methods and customized protocols. His deep expertise in good laboratory practices and good clinical practice has been instrumental in advancing research methodologies and ensuring compliance with international regulatory standards. A certified clinical research professional, Chris is an active member of the American Society for Testing Materials, or ASTM, and the Society of Clinical Research Associates, or SOCRA. He has made significant contributions to scientific literature, authoring numerous publications on antimicrobial efficacy and skincare product evaluations. His professional training encompasses advanced courses in data management, informed consent, and clinical trial regulations, reflecting his dedication to maintaining the highest standards in clinical research. We do encourage your questions for the presenter. You can submit them at any time in the questions box. Chris will try to answer as many as he can in the final section of our webinar. I will now turn the time over to our presenter, Chris Beausoleil.
Thank you, Chris. I hope everyone is having a great day. ASTM E1174 was published in 1987 to evaluate healthcare personal hand wash formulations, which was first defined by the FDA in 1978. The method and regulations have undergone several changes and modifications over the decades, with the most recent version of the ASTM method being released in 2021 and the FDA publishing a final monograph in 2017. This webinar will provide a historical overview of the procedures used to evaluate healthcare personal hand washes, with an in-depth look at the procedures and the current method and regulation, as well as considerations that must be made when performing such evaluation. I cannot start a discussion about hand washes performed by healthcare personnel without first acknowledging the work of Ignaz Philipp Semmelweis. He was a physician in Vienna, Austria in the 1840s, and in the 1840s, he noted infection rates in maternity wards were higher when medical students came directly from the autopsy room to examine patients. He recommended a practice of washing hands with the chloride of lime when leaving autopsy room prior to examining obstetric patients. At the time, mortality rate of childbed fever was as high as 18%. Following practices recommended by Semmelweis, that mortality rate was reduced by 2% and sometimes less. He published his observations and statistical results in the Etiology, Concept, and Prophylaxis of Childbed Fever in 1861. The evidence he published, along with numerous studies since, have supported the use of topical antiseptic products in medical care. In 1972, the commissioner of the FDA announced a review of OTC drug products for safety, effectiveness, and labeling by an independent council of experts. Also in 1972, the FDA issued a Federal Register notice requesting data and information for OTC antibacterial ingredients. This was a call for data from all topical antimicrobial drug manufacturers so that existing data could be used to assess claims of the products for the intended use. These notices resulted in proposed safety and efficacy requirements in 21 Code of Federal Regulations Part 333, topical antimicrobial drug products for over-the-counter human use. In 1974, the FDA established an advisory review panel on OTC topical antimicrobial drug products, the Antimicrobial 1 panel. This panel adopted multiple product categories, one of which was the healthcare personal hand wash, which was defined as a safe, non-irritating preparation designed for frequent use that reduces the number of transient microorganisms on intact skin to an initial baseline level after adequate washing, rinsing, and drying. If the preparation contains an antimicrobial agent, it should be broad spectrum, fast acting, and if possible, persistent. In 1978, the tentative final monograph was first published, presenting the tentative conclusions of the commissioner regarding safety, effectiveness, and labeling of topical antimicrobial drug products. This was based on their review of the final report of the Antimicrobial 1 panel. The published method was only 3 paragraphs. This is that section presented on this slide. In summary, the method involved artificial contamination of the hands by dipping the hands into a liquid culture or by spreading a known aliquot on the hands with a 1-minute wait. There was an option to also handle contaminated materials to simulate actual practice. Then use the product according to the label directions. This cycle of contamination, then washing, was to be performed 25 times with a minimum 5 minutes wait between each cycle. The wash water would be passed through a millipore filter to assay the microorganisms from the wash procedure. The hands are contaminated with a marker strain of microorganisms such as S. marcescens, a pigmented strain, or Bacillus subtilis, strain Bobigi. This original recommended procedure has changed significantly over the years. In the Federal Register issued on June 17, 1994, FDA published their 21 CFR Part 333 and 369 topical antimicrobial drug products for over-the-counter human use, tentative final monograph for healthcare antiseptic drug products. In section 333.470, subsection A to B2 is listed here, effectiveness testing of an antiseptic hand wash or healthcare personal hand wash presented new test procedures for this evaluation. The test procedures were identical to those published in ASTM E1174 1987, using S. marcescens as the indicator microorganism. I'll say S. marcescens, it's Ratio marcescens. Acceptance criteria for healthcare personal hand wash formulations was a 2-log reduction of the indicator organism on each hand within 5 minutes after the first wash, and a 3-log reduction of indicator organism on each hand within 5 minutes after the 10th wash. The method presented a statistical procedure for calculating the number of subjects required for this evaluation. And to validate the assay, the changes from baseline counts are compared to a control material at each sampling interval after washes 1, 3, 7, and 10. It is acknowledged that the reductions would be less variable, so comparisons could be made with a student's t-test or similar procedure. The Federal Register, published on May 1st, 2015, presented our 21 CFR Part 310, Safety Effectiveness of Healthcare Antiseptics, Topical Antimicrobial Drug Products for Over-the-Counter Use. It's a proposed amendment of the tentative final monograph and reopening of the administrative record proposed rule. This established new conditions for evaluating OTC healthcare antiseptics based on reevaluation of data requirements in the 1994 TFM due to comments received, input from public meetings, and independent evaluation by the FDA. In this publication, the FDA proposed the additional safety and effectiveness data are necessary to support generally recognized as safe and generally recognized as effective, grass or gray, determinations of OTC antiseptic active ingredients intended for use by healthcare professionals. This proposed rule does not address testing of final formulations proposed in the 1994 TFM. Part of this notification presented new criteria for effectiveness still based on bacterial log reductions. For healthcare personal hand wash active ingredients, proposed changes required data after just a single wash with demonstration of a 2.5 log reduction using a 70% success rate. The evaluation was no longer a 10-wash study, and more than 70% of the subjects tested must demonstrate greater than 2.5 log reduction after a single wash, which is a half log higher than the 2 log required in the 1994 TFM after a single wash. The actives should be different from a vehicle control and again must have adequate sample size to meet the requirements. On December 20th, 2017, the FDA issued their final rule for 21 CFR Part 310. This final rule was based on additional comments received after the 2015 proposed rule and continuing evaluation of the data. For effectiveness testing of the healthcare personal hand wash active ingredient, primary changes were the study design was changed to a 3-arm study with an active control and a negative vehicle control also being evaluated. Statistical analysis was changed to use of an average treatment effect after a single wash. The product must demonstrate superiority to the negative vehicle control by a 1.2 log 10 scale margin and be non-inferior to an active control by a 0.5 log 10 scale margin. Finally, it is required to have a minimum sample size of 100 subjects per arm, basically a 300 subject study. Now, concurrent with the FDA's work writing regulations for topical antiseptics, ASTM was also working on standardizing the test method. First E1174 method was published in 1987, which was similar to the procedures in the 1994 TFM. The method was updated in 1994 and again in 2000 with a title change to include consumer hand wash, which was then dropped from the title in the 2006 update. The method was updated again in 2013 with the most recent version being published a few years ago in 2021. In total, the method has been updated and revised 5 times since 1987. The subcommittee responsible for maintaining the method is ASTM E1174, the Antimicrobial Agents Subcommittee. The best way I found reviewing all versions of E1174 was to first look at the sections of the method. The current version consists of 18 sections. The original 1987 version had just 13 sections. I'll provide a brief overview of each section in the current 2021 version and how that section has changed over the revisions. Starting with the scope of this method in section 1, the method is to determine the effectiveness of antimicrobial hand wash agents to reduce transient microflora, skin flora, and you must have knowledge of microbiological techniques to use this method. In 2000, knowledge of regulations pertaining to protection of human subjects were added to the scope. These are 21 CFR Part 50 concerning informed consent and 21 CFR Part 56 for an institutional review boards. Subjects should voluntarily provide informed consent to participate in the study, and the study should be performed under the purview of an IRB or an IEC, an independent ethics committee. Essentially, a researcher should follow current good clinical practices when following this method. The method does not address all safety concerns, and those are the responsibility of the user. I'll address some of these in this presentation. The reference documents listed in section 2 has changed little over the years. The one consistent reference document is E1054, which is currently titled Standard Practices for Evaluation of Inactivators of Antimicrobial Agents. This method outlines how to validate inactivators or neutralizers used in this method that will stop the antimicrobial activity of a product at a specific sampling time. This method requires its own webinar, so I'll not go into much detail about how it is used. In 2013, E2755, test method for determining the bacterial eliminating effectiveness of hand sanitizer formulations using hands of adults was added as a testing alternative to evaluate waterless hand rub products. Similar to E1174, with one of the main differences being the use of a small volume of a challenge inoculum, a dry contamination, we call it, versus the wet contamination of E1174. In 2021, E2756, terminology related to antimicrobial and antiviral agents, was added. This document had been in development for years by the E3515 committee to standardize the terminology used between all the methods developed and maintained by the subcommittee. Terminology was added in 2000. Terms specific to this standard are active ingredient, substance that inhibits or inactivates microorganisms; healthcare personal hand wash, reduces transient microorganisms on the hands; resident microorganisms, that's the normal microbial flora that resides on the skin; test formulation, formulation with an active ingredient; test organism, an inoculum applied to the hands that is easily identifiable and simulates transient microbial contamination of the hands; and transient microorganism, organisms from the environment that contaminates but do not normally colonize the skin of the hands. Most terminology for this method is addressed in the E2756 terminology method. The summary of the methods that will be used are that the study involves volunteer subjects who have refrained from antimicrobial use for 7 days. This pre-test conditioning period is important so that subjects don't participate in testing with any residual chemicals, soaps, compounds on their hands that would have an effect on the test organism being applied to the hands. A broth culture of S. marcescens is used to contaminate the hands. In the 1987 version, the method required measurements of efficacy performed after washes 1, 3, 5, 7, and 10. In the 1994 version, E. coli was added as an alternate test organism. This organism has not been included in the FDA regulations. We have used this organism when evaluating products primarily intended for food service industry. In the 2000 version, measurements performed after a single wash and 10 additional washes were added, and the method now required use of sampling fluid that incorporates a neutralizer for all post-wash samples. Prior to this, neutralizer was optional for the first wash. In 2021, measurements were changed to following a single wash with an appendix added describing the alternate hand contamination recovery procedure similar to the changes for the additional washes made in the 2000 version. Finally, due to the use of the bacterium to contaminate the hands, researchers should be aware of potential health risks from its use and take precautions. I'll discuss these later. The significant use section in the first 87 version was simply just testing the degerming effectiveness of antimicrobial hand wash agents, agents used by healthcare personnel intended for frequent use and intended to reduce the level of contamination acquired through contact with contaminated objects or people. Since 2006, the section has been simplified to the method is to be used to test antimicrobial effectiveness of hand wash formulations, formulations designed for frequent use and to reuse transient bacterial flora on the skin of the hands. The current 2021 version added that waterless formulations should be added using that E2755 test method that I previously listed. The apparatus in this section 6 has changed little since first published in 1987. Required equipment for performing this procedure are: you need a colony counter, such as a Quebec colony counter to help with counting the bacterial colonies. S. marcescens is very easy to count. The colonies are deep red and easily distinguishable from any other colonies that may be present on the plates. You'll need an incubator for incubating the plates. Serratia marcescens requires a 25-degree incubator, which is best temperature for producing that red color. If using E. coli, you'll need a 35-degree incubator. As with any microbiology study, you'll need a sterilizer to sterilize all the media needed and sample collection tubes and bottles you'll be using. There are numerous time steps in this procedure to be performed, and a timer will be needed to track those procedures. This is a hand washing method, so you'll need a hand washing sink with water faucets at a height to allow hands to be held higher than elbows during washing. Our sink that we use at Nelson is a large basin with multiple adjustable height faucets. The sink will also need a top water temperature regulator so that the water is a constant 40 degrees plus or minus 2 for the washing procedures. Now, the section should also state that that wash sink should be able to provide a water flow of 4 liters per minute, which was added to the procedure in section 10 in 2006. I expect this to be added in the next versions of this method. The section for reagents and materials is changed since 1987. You'll need graduated pipettes for the inoculum and sample collection and adjustable volume pipetters for diluting and assaying the samples. Water dilution bottles listed are somewhat dated, as there are other options available that are less cumbersome and expensive. The glass dilution bottles were used to hold the 75 mL of sampling fluid, which is then sterilized for use for the sampling procedure. When I first started in 1994, we quickly discovered that this was difficult to do and not consistently lose volume in the bottles during sterilization, resulting in less than 75 mL remaining in the bottles. We now first prepare and sterilize our sampling fluid in bulk and then aseptically dispense 75 mL in sterile plastic containers for use during sampling. The cleansing wash changed in 2006 with directions for preparing a soft soap solution to use so that all studies could be performed using the same soap for cleansing. Test material is or are the antimicrobial formulations that are being evaluated. For sampling, sterile gloves have always been part of the method. Plastic bags were added as an alternative in 1994. We have used both, and I'll discuss them both in detail later in this presentation. The sampling solution is a buffered solution with a mild detergent and neutralizing additives added to inactivate the antimicrobial activity of the test materials. Diluting fluid is sterile Butterfield's buffered water or other diluent, including neutralizing additives. Agar that is used to plate the samples should be a soybean casein digest agar for Serratia marcescens for E. coli. The method suggests a mug agar. However, we have had success using McConkey agar, which also allows for differentiating E. coli colonies. All these media need to be properly validated, be non-toxic to the challenge microorganism, and effective at neutralizing the antimicrobial ingredients in the test materials per ASTM E1174. The broth is used to prepare the test microorganism. This is soybean casein digest broth, but can be another liquid medium validated to support growth of the test of the organism. In 2021, the materials needed for the decontamination of the subject's hands after testing were added. These are 70% ethanol for a 1-minute rinse, 4% CHG skin cleanser for a 4-minute scrub wash, and a triple antibiotic ointment to be applied after the rinse and scrub wash. You should consult with a physician regarding the use of the ointment. Also, the ointment should only be used once by subjects under supervision, and the subjects should be advised to not apply ointment on their own after leaving the lab. Overuse of the ointment by the subjects could cause skin reactions. As previously mentioned, the test organisms in the challenge suspensions used to contaminate the hands are Serratia marcescens, ATCC number 14756, or Escherichia coli, ATCC number 11229, which was added in 2000. Prior to 2000, a suspension was created by inoculating a 2-liter flask containing 1 liter of soybean casein broth with a stock culture of the organism. In 2000, instructions were added that the stock culture should be at least 2 24-hour soybean casein broth cultures, but no more than 4 transfers from the original ATCC culture. If using a source other than ATCC for your stock culture, you should confirm that the number of transfers from the ATCC culture is less than 4. From the stock culture, inoculate the final test suspension with a stock culture using a ratio of 0.1 mil of stock culture for every 100 mils of broth, or 1 mil of stock culture into 1 liter of broth. The ratio was added if a researcher needed less than 1 liter of the suspension for their testing. A final test suspension should be no more than 5 transfers from the original ATCC culture. Incubate the broth cultures for 24 plus or minus 4 hours at the appropriate temperature for the organism. If testing over multiple days, try to prepare the suspensions the same for each test day. When used for testing, swirl or shake that test suspension before withdrawing it to contaminate the hands of the subjects. The suspension can be used up to 8 hours. Assay the culture at the beginning and end of the use period, and the population of the culture should not change by more than half a log. Our S. marcescens cultures are typically around 1 times 10 to the 9th CFUs per mil, while our E. coli cultures are around 5 times 10 to the 8th CFUs per mil, and they change little over the 8-hour use periods. We recommend preparing isolation streaks of the stock cultures used to make your flask to check for culture purity, and gram stain the test procedure, the prepared test suspension prior to use on test day. Prior to 2000, Section 9 regarding subjects recommended to complete testing with 12 subjects. This was the last time a sample size was listed. Based on the statistical test you are performing, you can calculate sample size needed for your study. You need to recruit healthy human volunteers. This includes providing them with an informed consent form following the regulations in 21 CFR Part 50, describing the purpose of the research, the requirements for the subject, the procedures that it will perform, and anything else so that the subject is fully informed and understands the study they are volunteering to participate in. I recommend you recruit more subjects than what will be needed because subjects will withdraw or be disqualified prior to or on test day due to cuts in the skin, hangnails, scratches, or something else. I have seen some unusual disqualifying conditions over the years performing this study. The subject should have no clinical evidence of dermatosis, open wounds, hangnails, or other conditions. You should consult with a physician regarding additional inclusion and exclusion criteria such as medical conditions, allergies, etc. We work with consulting physicians who regularly update our criteria. When recruiting the subjects, instruct the subjects to avoid exposing their hands and forearms to antimicrobial products, antimicrobial containing products, medicated soaps, shampoos, lotions, acids, bases, and solvents, basically irritants, household chemicals, bathing in biocide-treated pools, hot tubs, or spas. Provide the subjects with a kit containing non-antimicrobial products to use for at least 7 days prior to the test day. Include rubber gloves to be worn when contact with items to avoid cannot be avoided. This helps to standardize the condition of the skin for all subjects and prevent any interference caused by exposure to any antimicrobial agents residing on the skin on test day that may interfere with the test organism prior to using the test materials. The procedure has changed significantly over the years. The first was the volume of the test organism suspension used to contaminate the hands. Was changed from a single application of 5 mL of broth suspension to 3 1.5 ml aliquots of a suspension for a total of 4.5 mL used. The procedure was changed because 5 mL is a lot of fluid to be added to the hands so that such that the subject does not lose any during spreading of the broth over the hands. This could result in a huge variation in the recovery data. In 2000, the procedure was changed to a 3-step procedure. Step 1, 1.5 ml suspension is added to the subject's cupped hands, one hand cupped over the other, and then spread over the hands up to the wrist for 20 seconds, followed by a 30-second air dry. This is repeated for step 2. For step 3, the same volume is applied again, but the air dry is performed for 90 seconds. The hands will still be damp, wet after the 90-second air dry. This 3-step procedure reduced loss of the broth by the subjects during spreading over the hands and decreased the variability in the recovery populations. Another change in the procedure was the number of washes performed. This has changed from 10 washes to a single wash evaluation, or 25 from the first TFM in 1978 to a single wash. The reason for this change was that the continued effectiveness was not considered necessary, and the FDA agreed in 2015 that the critical element of effectiveness is that a product must be effective after the first application because that represents the way in which the healthcare personnel hand washes are used. You should follow standard biosafety procedures for your facility. We have our subjects wear disposable aprons and face shields to protect them from the test materials and inoculum while participating in testing. Any materials touched by the subjects are used for testing or disposed of following our standard biohazardous waste procedures. The procedure can be summarized into 5 steps. First, the subjects perform a cleansing wash with soft soap. Subjects wet their hands, then 5 mL of soft soap is added. The subjects wash their hands in the same manner as to be used for the test formulation wash for 30 seconds, then rinse their hands in a similar manner, which is typically performed for 30 seconds. Subjects finish with lightly pat drying their forearms and hands using a separate paper towel to start pat drying from the forearm up to the hand, and then repeating with the other hand with a new paper towel. This step removes the dirt and oil from the hands and helps to familiarize the subjects with the wash procedure. During this step, we also perform, prior to doing the cleansing wash, we also perform a practice contamination with the tap water to also help familiarize the subjects with the hand contamination procedure before the wash. Second step is the subject's hands are contaminated with and then sampled for baseline recovery. This baseline recovery is then followed by another cleansing wash performed as before for step 3. For step 4, the hands are again contaminated and then the subject performs a wash with the test formulation, which is then followed by a sample for post-wash recovery from both hands. After the post-wash sample, subjects then complete a hand decontamination procedure with ethanol, chlorhexidine scrub wash, and application of the antibiotic ointment. In the appendix of the method, there's an alternate hand contamination recovery procedure for evaluating test formulation's efficacy after a single wash and then after 10 washes, 11 total washes performed. After the first wash, the subjects performed 10 additional hand contamination washes with a sample taken after that final wash. This was included to allow researchers to also evaluate residual or persistent antimicrobial effects of a hand wash when repeatedly challenged. The wash and rinse procedure was split out from the procedure section, section 9.0, in the 2000 version. Section 11.1 states to conduct the test in accordance with the use directions for the test material. If there are no instructions for liquid formulations, leave-on, and solid formulations to be used are provided. All wash procedures should start within 10 seconds after completion of the 90-second air dry for the hand contamination. For liquid formulations, 5 mL of test material is first dispensed in the subject's cupped hands, followed by them spreading the material over their hands and lower third of the forearms. Have the subjects then briefly wet their hands less than 1 second. One thing to note here is that with this wetting step may dilute or rinse off some of the 5 mL of applied formulations. It's not uncommon to switch the order to help maintain the volume of test material on the hands and to reduce any variability in the amount remaining on the hands for the wash. This should be described in detail in your test protocol. After formulation is applied and the hands are wet, subjects will wash in a vigorous manner for 30 seconds. To standardize the wash used by subjects, we have adapted the standard wash procedure from EN 1499, 2013, which is also the World Health Organization's recommendation for performing a hand wash. It is a 7-step procedure. So we have the subjects rub palm to palm, then right palm over the left dorsum, and then left palm over the right dorsum. Palm to palm, fingers interlaced, back of fingers to opposing palms with fingers interlocked. Rotating rubbing of the right thumb clasping the left palm and vice versa. We describe this as revving the motorcycle. Rotational rubbing backwards and forwards with clasped fingers in the right hand in the left palm and vice versa. This focuses on the fingertips, nails, and cuticles. And then at the end, rotational rubbing of the forearms above the wrist. This whole procedure, when you we'll talk a subject through it, will take about 30 seconds to complete. After washing, the subjects then rinse their hands. What we describe internally here as passive rinsing was added to the method in 2006. During the 30-second rinse, arms are held over the sink, flexed upright from the elbows and not touching the sink. Subjects rinse from elbows up to the hands, alternating between hands and avoiding touching hands. Studies performed prior to 2006 and studies with use instructions provided that include a thorough rinsing have involved what we call active rinsing, allowing the hands to touch to remove the test formulation thoroughly when rinsing the hands. The method of rinsing should be detailed in your study protocol. The method still has a step following the rinse to lightly pat dry the hands and forearms with paper toweling, followed by a small note that hands are not to be dried if sampling is to be performed. Subjects should hold their hands upright until sampling procedure is performed. I think this may be updated in the next revision to clarify that clarify the post-rinsing step, as it should be, as it is confusing to list pat drying first, followed by that small note. The hand should be gloved wet following the rinse. The recommended instructions for leave-on formulations are apply 3 mL of test material dispensed in the subject's cupped hands, who then spread material over their hands and lower third of the forearms. Have the subjects continue rubbing in a vigorous manner for 30 seconds. We use the rubbing procedure similar to that we use for the liquid hand wash formulations. It is noted that you really, again, should use E1174 instead of this method. E1174 is important because waterless products are typically used on non-visibly soiled hands, and the low volume used for the contaminating the hands helps to replicate this use. For solid formulations or bar soaps, have the subjects wet their hands and forearms, then have the subjects wet the product. Subjects then rub the product between their hands and on their forearms for 15 seconds. The product is then set aside. The subjects then lather the lower third of their forearms and hands for 30 seconds. The subjects can add water to the hand if the hands start to dry. The standard wash procedure can also be used here as well. Subjects then rinse their hands and forearms for 30 seconds using the passive rinse procedure. Subjects' hands and forearms are lightly patted dry with a paper towel. There is no note here for not drying the hands before sampling. I do expect this to also be addressed in the next revision. I'll note again here that the materials used in the study can be hazardous, and you should dispose of all materials used according to standard operating procedures. This is all everything that's used in the lab, including the paper toweling that the subjects are using to dry their hands after all the test procedures. The bacterial recovery is based on the glove hand method for sampling that was first published in July of 1972. Here's a reference to that article. The sampling procedure was developed to evaluate the effect of antimicrobial agents on normal resident microbial flora of the human skin of the hand. This method was originally included as part of the method for evaluating surgical hand scrub products in the 1978 TFM. In the 1994 ASTM method, low bioburden plastic bags were added as an alternative to gloves. In 2000, this procedure was split out into section 12.0. For baseline samples, we use a sampling fluid with no product neutralizers. We do this so that there are no residual neutralizers left on the hands prior to product use. For all post-wash samples, use sampling fluid with product neutralizers included in the glove. When using gloves, within 1 minute of completing a wash, place loose-fitting, unlined, powder-free gloves onto the hands. The method suggests a latex glove to use. It can be difficult to source gloves. We have found it's very difficult to source gloves sometimes intended for healthcare use with manufacturing delays, and especially after the recent pandemic. We have found that there are many types of new sterile gloves being offered, and have found that the new sterile nitrile gloves for clean rooms work well, are non-antimicrobial, and are lower in price. Most gloves that we have tested for medical use, latex or synthetic, have had some antimicrobial properties and can be difficult to source and are considerably more expensive. Within 1 minute of gloving, add the sampling fluid, then secure the gloves above the wrists. We use an elastic tourniquet to secure the gloves. Uniformly massage all surfaces of the hand for 1 minute, paying attention to the fingers and flipping at 30 seconds. The flipping was added in 2006. When we first developed our SOP for glove use sampling in the 90s, we did not flip the hands because we uniformly massage all aspects of the hands and fingers throughout the 1 minute procedure using both of our hands and fingers. This has been described by our new analysts or has been compared to playing video games. So some of them are actually pretty good at doing glove juice procedure. After completing the massage, aseptically retrieve 5 to 10 mils of the sample and place into a sterile test tube for assaying. For the bag juice procedure, place low bioburden plastic bags onto the hands. All the bags we have used have had low bioburden when tested. You can test the bioburden by first adding sampling fluid to the bag, massage the fluid in the bag for approximately a minute, and then spread plate 0.1 mil of the fluid from that bag. After the bags are on, have the subjects hold the bag while retrieving the sampling fluid. If they don't hold that bag, it's going to drop off their hands when you add the sampling fluid. Secure the bags above the wrist, trying not to capture too much air in the bag. Uniformly massage all surfaces of the hand for 1 minute, paying particular attention to the fingers, and flip the hand after 30 seconds to thoroughly massage all aspects of the hand. This is an important step when using the bags. As you can see, the fluid will pool in the bottom of the bag. In the glove, the fluid fills in the fingers and the lower part of the hand. Flipping allows the sampling fluid to be rubbed on all aspects of the hands in the bag. After massaging, aseptically retrieve 5 to 10 mils of the sample. Enumeration of the samples has been consistent since 2000. Just use standard microbiologic techniques to assay and enumerate the number of S. marcescens or E. coli in the recovered samples. Samples should be processed soon after recovery to prevent propagation or reduction of organisms in the sample. This whole time should be validated using the methods in E1174 when validating the media and neutralizers. We process our samples within 30 minutes of collection. We do not refrigerate or freeze samples for processing later, and any such process should be properly validated to verify the microbial populations in the samples do not change. For serration marcescens, dilution of the sample should be plated using surface plating methods so that the red pigment can develop during colony growth for counting. Colonies in agar will not develop that red pigment. 3 plating methods you can use are membrane filtration, which is a very expensive and time-consuming method. Spread plating, you plate no more than 1 ml of the aliquots of the sample, or spiral plating, which requires a commercially available spiral plater such as the Whitley Wasp Touch, which is what we use. Incubate the plates for 48 plus or minus 4 hours at 25 degrees, and then use standard counting methods, using standard counting methods. You can use a validated computer imaging system to count spiral plates. Here is an example of one of those plates from one of our studies. And you can see the bright red that the S. marcescens grows as. For E. coli dilutions of the sample can be plated using membrane filtration, pore plating, or spread plating if using the mug indicator. That media requires a UV light to count the fluorescent colonies when counting. As I mentioned earlier, we use McConkey agar. We spread plate 1 ml aliquots. The E. coli produces pink colonies on McConkey agar, which are easily counted. Incubate the E. coli plates for 18 to 24 hours at 35 degrees, and then count using standard counting methods. Section 14.0 for hand decontamination is the most important step of this method for safety of the volunteer subjects. If hands aren't fully decontaminated, they may develop infections on the skin due to contaminating microorganism. Up until the 2021 method, the suggested procedure was to just scrub with 70% isopropyl, propanol or equivalent. We were conducting many of these studies since 1994, and this decontamination process was not sufficient to ensure the safety of the subjects due to the number of adverse events we were observing. We incorporated a chlorhexidine washed antibiotic ointment in the early 2000s and saw a significant reduction in adverse events. The procedure in the 2021 method is similar but not identical to our procedure. The 3-step procedure consists of 1, subjects rinse their hands, forearms, upper arms up to the elbows for 1 minute with 70% ethanol. Following this rinse, subjects' hands and forearms are allowed to fully air dry before proceeding to the next step. For step 2, subjects perform a supervised 4-minute wash with 4% chlorhexidine gluconate hand wash. Method states to have subjects use a scrub brush for the first minute. We do not use a scrub brush because of the risk of creating microabrasions in the skin. We also caution subjects about any potential reactions they may have from use of the 4% chlorhexidine gluconate and to seek medical attention if they do experience any kind of a reaction to it. Subjects apply a topical antibiotic ointment to the hands. We only use ointment for studies with S. marcescens. We do not use it for studies using E. coli. This has not been required in our experience. You should consult with a physician regarding the use of the ointment. Now, brace yourself, math is coming. Prior to 2000, section 15.0, determination of the reduction simply stated to calculate the changes from baseline counts obtained with the test material at each sampling interval. In 2000, additional details for the determination were added. First, convert the plate counts to log 10 CFU hand values. This is our formula for converting plate counts to log 10 CFU hand recovery values, or R values. Multiply the volume of sampling fluid by the average CFU count observed on 2 or 3 plates times 10 to the power of the plated dilution as a non-negative integer. Using those calculated R values, you then calculate the average of the right and left hand R values at each sampling interval. Simply just add the right and left values and divide by 2. Using those R values, determine the reduction at each recovery interval, subtract the post-wash average R value from the average baseline R value. While not part of the method, we have also reported percent reductions. To determine percent reduction, subtract the antilog of the average post-wash R value from the antilog of the baseline R value, divide that by the antilog of the average baseline R value, and multiply by 100. The section for comparison to test material has changed little since 1980. If comparing test materials, we recommend testing equal number of subjects for each material. Number of subjects required will depend on the statistical test being used and the regulatory requirements. Remember, in 2017, the FDA recommends a minimum of 100 subjects for testing active ingredients. Assign subjects to each material randomly. There are many statistical programs available that will generate assignments. You may need to consider stratifying the randomization by sex to ensure that a ratio of male to female subjects isn't skewed towards one product versus another. Test parameters should also be equal for all products. Simply just follow the procedures outlined in this method for all subjects. Test materials should be tested at the same time. We test approximately 6 subjects every hour with a maximum of 24 to 30 subjects a day. A balanced randomization where products are assigned equally within blocks of subjects will ensure that the test materials are being tested at the same time. For example, if just testing 2 products, randomization block of 4 will assign 2 subjects to each product for every 4 subjects tested. Prior to 2021, a precision and bias statement could not be made for the method. In the current version, the results of a reproducibility and repeatability study were presented for a statement of precision. For baseline controls, the overall mean was 9.12 log with repeatability SD of 0.25 and reproducibility SD of 0.29. We see similar values and expected to have approximately and expect to see 9 logs on the hands with low variability when following the procedures and method. If unusually low values observed, 8 logs, let's say, or lower, for example, subjects may have not avoided antimicrobials or some inoculum was lost during the contamination step. Higher values could indicate an error using the inoculum or an error during the assaying of samples. For a wash with a chlorhexidine product, the overall mean log reduction was 2.69 with repeatability SD of 0.46 and reproducibility SD of 0.49. With the use of the product, variability will increase and will depend on the subject's ability to follow directions during the wash, the formulation of the product, and the wash and the rinse procedures performed. To reduce bias, there is no accepted reference value, and best practice is to incorporate randomization to balance the use of the products by subjects, or balance the use of the products by the subjects by group, day, and sex. I'll conclude with some of my final thoughts. There's one final section, section 18, which lists keywords. This is useful when performing searches or queries. This section is not relevant for use in the method. E1174 will be reevaluated again in 2026. I expect there to be some discussion about minor changes or added clarifications to be added, some of which I've already noted. FDA is still accepting comments regarding the published requirements for healthcare personal hand wash active ingredients. These are the relevant dockets for those comments and can be searched on the FDA's website. A recent publication presented interesting results evaluating the use of plastic bags versus gloves and discovered hand size may also be a factor to consider when conducting this evaluation, especially when using gloves. This was a study published by Moyer et al. In American Society of Microbiology's Microbial Spectrum. Here's the reference for that study. Finally, we have recently started using bioinstrumentation to measure skin moisture content to help in admitting subjects with normal, healthy skin. There are devices that measure skin moisture content and other skin attributes that can be used to verify healthy skin and may prove to be helpful in the future. Thank you for your time today. I'll now try my best to answer any questions that you may have.
Yeah, thanks a lot, Chris. It was really insightful. We do have a few questions. I will go ahead with the first question. Are current FDA criteria achievable by FDA-approved products already or currently on the market?
I assume that's referencing the ATE evaluations. And for the non-inferiority, yes, but the superiority, there might be some issues with it. If you see in the 1994 monograph, when it was first published, when products were being evaluated, they just had to achieve a 2-log reduction after the first wash. Now, just general hand washing, if you read some publications, that will generally get you about a 1.5-log reduction. And if products were approved with that 2-log criteria, that means they only had greater than a 1/2-log reduction, or their only difference by a 1/2 log. That ATE superiority requirement is 1.2 log reduction. So products would have to have greater than a 2.7 log reduction after a single wash in order to meet that requirement. Some products may have difficulties meeting that requirement if being reevaluated. Previously approved products may have some difficulties meeting that if being reevaluated using that procedure.
Okay, thank you. Just a reminder to our audience, if you're not able to ask any of your questions or get answers to your questions during this live webinar, you can reach out to Chris directly through his email address that is listed on the screen, cbeausoleil@nelsonlabs.com. I'll go ahead with the next question. Do you believe N equals 100 will be required for future monograph actives in the clinical studies?
I don't know. 100 for a healthcare personal hand wash study is more than enough subjects for basically any statistical test you want to use to analyze the data. The variability from the study, as mentioned before, is pretty low if you follow the procedures in this method. And in order to determine equivalency, superiority, non-inferiority, or a significant difference, 100 is more than what is actually needed for those statistical tests. But I don't know if that's going to continue to be a requirement going forward.
Okay, an honest answer. It looks like this is our final question. Do you have any idea when we might see a final monograph for formulated products?
I don't know. It's interesting that in 2015 and 2017, it's noted that those regulations are for active ingredients. I may have to just do some more research and work into it, and ultimately it's going to come down to, I mean, if anybody's doing any kind of, or trying to get their product approved, we recommend everybody should just work directly with the FDA and work with them to make sure that they're providing them with the data that they need to make their assessment. But again, I don't know. I use the methods, and yeah, moving forward, I don't know what's actually going on with the regulation, or what will end up happening with the regulation. I don't feel comfortable predicting that.
Okay, well, we appreciate that. Well, thanks again to Chris Beausoleil for a thorough and insightful presentation. And again, if he was not able to address your question during the presentation, he will be happy to address it after we conclude. Reach out to him directly.
Yes.
His contact information is listed there. I'd like to thank you all for taking part in this live session, and I really hope you found it helpful and well worth your time. And I hope you enjoy the rest of your day.